Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine this scenario. You have a cancer treatment, one that's working, and then suddenly it just stops.
Yeah, it's a really tough reality for many people with estrogen receptor-positive breast cancer, a real roadblock.
But maybe there's a way around it. We're looking into some really interesting new research today that might offer a potential detour.
That's right. We're dealing with a big challenge in treating the most common type of breast cancer. About 70% of cases are estrogen receptor positive.
Meaning their growth is fueled by estrogen.
Exactly. So the first line of attack is often hormone therapy to, block that estrogen or stop its production.
But the cancer can become resistant.
It can adapt, find ways around the blockade. That's the resistance problem we're talking about.
So today, we're diving into a study published in RSC Medicinal Chemistry. It looks at some completely new compounds.
Designed specifically to tackle this hormone therapy resistance in estrogen receptor-positive breast cancer.
And the really clever part, it seems, is that they're targeting two key enzymes involved in making estrogen, not just one.
Yes, they're going after both aromatase and steroid sulfatase simultaneously.
Okay, so we have a health news article summarizing this from April 9th, 2025, and the actual scientific paper itself.
Yep, the paper's titled Development of Benzofuran-Derived Sulfamates as Dual Aromatase-Steroid Sulfatase Inhibitors, and so on. Design, synthesis, evaluation.
Right, and our aim here is to pull out the key takeaways, make sense of this potentially exciting development without getting bogged down in the super technical stuff.
We want to understand how these compounds work, why hitting two targets might be better, and what this could mean down the road.
Okay, so let's start with the basics. Why does this resistance happen? You mentioned the cancer adapts.
Well, yeah. Estrogen receptor-positive breast cancer needs estrogen to grow. Current therapies often target aromatase, an enzyme that makes estrogen from androgens.
And that works well initially.
It can, yes. It starves the cancer cells.
Right.
But over time, the cancer can find other ways to get estrogen. It effectively bypasses that aromatase blockade.
Which leads to resistance. So this new approach targets aromatase and this other enzyme, steroid sulfatase. Why both? What's the logic?
The thinking was if you block estrogen production via two different routes at the same time, you might have a better shot at really cutting off the fuel supply.
Even if the cancer tries to compensate using one pathway.
Exactly. So aromatase is one target, steroid sulfatase is the other, and steroid sulfatase is actually quite interesting.
How so?
It converts inactive estrogen sulfates, which are just floating around, into active estrogens that fuel the cancer.
So it's like activating a reserve supply.
Kind of, yeah. And the paper notes that steroid sulfatase actually produces about 10 times more of one type of estrogen, estrone, compared to aromatase.
10 times, wow. Okay, so blocking both seems crucial then. That's the hypothesis. A much more comprehensive shutdown of estrogen production. Think of it like trying to empty a sink with two faucets running. You need to turn off both.
Good analogy. Right, so how did they actually make these dual-action compounds? What was the process? So the team designed and then synthesized these new molecules, which they call dual aromatase-steroid sulfatase inhibitors, or DASIs.
Okay.
They used a common chemical structure, benzofuran, as a starting point, like a basic framework.
Was it straightforward? Did the first tria work?
Oh, not quite. It never is, really. Their first attempts used a different structure based on something called a triazole.
And how did those perform?
They were actually pretty good at inhibiting aromatase. They blocked that enzyme well.
Well,
But they had basically zero effect on steroid sulfatase. It was like a key fitting only one of the two locks they needed to open.
okay, so 1 out of 2. Did they figure out why?
They used computer simulations, molecular dynamics studies to kind of see how the molecules were interacting.
Right, virtual experiments.
Yeah. And the simulations suggested that the triazole part might just be too bulky, too large to fit properly into the active site of the steroid sulfatase enzyme.
Like a physical mismatch. Steric hindrance, they call it.
Exactly that.
Wow.
It just couldn't bind effectively to do the job on steroid sulfatase.
Okay, so that approach didn't quite work. What did they try next?
They shifted to a different type of compound, benzofuran ketone sulfamates.
And these?
These had the opposite issue. They were quite effective at inhibiting steroid sulfatase.
So they got the second lock open?
Right, but they didn't really inhibit aromatase much at all, still just one target hit effectively.
So close, yet so far. It sounds like they were learning piece by piece. What was the breakthrough?
This is the really fascinating part. They made what seems like a tiny chemical change to that benzofuran ketone sulfamate structure.
Tiny how?
They added a methyl group, just one carbon atom with 3 hydrogens at a very specific spot on the benzofuran ring. Position 3, technically.
And that small tweak did what?
It had a massive effect. Suddenly, the compound showed significant inhibitory activity against both enzymes, aromatase and steroid sulfatase.
Wow. Just adding that small group flipped a switch.
It seemed to. It was the key adjustment needed to make the molecule work on both targets. The news article called it an unexpected but crucial insight.
Incredible. Were there specific compounds from this group that looked particularly good?
Yes, 2 stood out in the lab tests. They're labeled 19B and 19E in the paper. One has a chlorine atom added, the other has a methoxy group. Subtle differences, but both showed that optimal dual inhibition.
Okay, so they designed them, made them, and saw promising signs. Next step, testing them in a biological system.
Exactly. They needed to see if they worked in actual cells. They used JG3 cells.
Why those?
They're known to have high levels of both aromatase and steroid sulfatase, so they're a good model system for this kind of test. They basically treated these cells with their different compounds.
And what happened? Let's start with those early triazole ones.
As expected, the triazole sulfamates showed good aromatase inhibition in the cell's low nanomolar IC50 values, meaning very potent.
IC50 meaning the concentration needed to inhibit 50% of the enzyme activity.
Correct. Potent for aromatase. But again, virtually no steroid sulfatase inhibition. They needed really high concentrations, over 10 micromolar, to see any effect there. Very weak.
Any surprises with those?
Actually, yes. Some byproduct molecules formed during the synthesis, related carbamates, turned out to be even more potent aromatase inhibitors than the triazoles they were initially aiming for.
Huh. Interesting side discovery.
Yeah.
Okay, what about the ketone sulfamates, the ones that hit steroid sulfatase but missed aromatase initially?
In the cell tests, those ones without the crucial methyl group showed pretty weak aromatase inhibition, IC50s over 1 micromolar.
So not great for aromatase in cells.
No. But as designed, they showed good sub-micromolar steroid sulfatase inhibition in the cells. Confirm that part work.
And then the ones with the added methyl group, the potential stars.
Yes. This was the confirmation they needed. The ketone sulfamates with a methyl group like 19B and 19E demonstrated the dual inhibition they were aiming for.
Can you give us the numbers again for those?
Sure. Compound 19B had an aromatase IC50 of about 137 nanomolar and a steroid sulfatase IC50 of 48 nanomolar, both good.
And 19.
Even stronger on aromatase, only 35 nanomolar IC50. And still good steroid sulfatase inhibition at 164 nanomolar.
So clear evidence in a cell system that adding that tiny methyl group unlocked the dual activity.
Absolutely. It really drove home how critical that specific structural modification was.
Now, you mentioned the computer simulations earlier. Did they go back to those to understand why the methyl group made such a difference?
They did. The simulations helped visualize how the molecules were interacting with the enzymes at an atomic level.
What do they show for aromatase?
For aromatase, the original triazole compounds bound using a nitrogen atom in that ring, making hydrogen bonds. But the simulations supported the idea that the ring's bulkiness likely caused that steric hindrance with steroid sulfatase.
Okay. And the ketone sulfamates. How did the methyl group change things there, according to the simulations?
Well, without the methyl group, the ketone sulfamates seemed to bind a bit indirectly to the important iron center in aromatase, maybe via a water molecule. That could explain the weaker inhibition.
And with a methyl group?
Adding the methyl group, like in compound 19E, seemed to allow the molecule to snuggle in differently, achieving a more direct and likely stronger interaction with that iron center.
So a better, more direct fit, leading to better inhibition.
That appears to be the case, based on the modeling. It helped explain the improved aromatase blocking.
And what about steroid sulfatase? How did the simulations show those compounds working?
For steroid sulfatase, the simulations indicated that the benzofuran sulfamate part positioned itself nicely in the active site.
How so?
The benzofuran part nestled into a more sort of oily, lipid-friendly pocket, while the crucial sulfamate group sat in the watery catalytic region.
And that sulfamate group did the blocking.
Yes, it formed hydrogen bonds with key amino acids essential for the enzyme's function. It basically jams the works, preventing the enzyme from grabbing its normal target, estrogen sulfate, and activating it.
Okay, so the simulations nicely backed up the lab result and explained the why behind the success of the methyl group addition. It helped achieve effective binding to both enzymes.
Exactly. It connected the structural changes to the biological activity they observed.
Right. So let's pull back and look at the bigger picture. What are the major implications of this work? If this holds up, what could it mean?
The biggest potential is clearly a new type of hormone therapy, one specifically for estrogen receptor-positive breast cancer that stopped responding to current treatments.
By hitting both estrogen production pathways harder.
Precisely. By inhibiting both aromatase and steroid sulfatase, these dual inhibitors could potentially lower estrogen levels much more effectively within the tumor environment.
Which might overcome resistance.
That's the hope. It could potentially resensitize the cancer or just be more effective in patients who have already developed resistance. It's, a potential new tool in the toolbox.
And the finding about the methyl group itself, that small change having such a big impact, that's significant too, isn't it?
Absolutely. It's a fantastic example of how subtle changes in molecular structure can dramatically alter biological activity. That unexpected finding was key.
It really highlights the precision involved in drug design.
And sometimes the serendipity. It underscores how much we can learn from careful chemical modification.
Does this also open up benzofuran-based molecules as a whole new class to explore for this purpose?
It certainly suggests that. The study demonstrates that this benzofuran scaffold is a viable starting point for creating these dual aromatase-steroid sulfatase inhibitors. It could spark more research using this type of structure.
This all sounds very promising, but we should also talk about the limitations, right? This is still early days.
Oh, absolutely. Very important point. This is preclinical research. All the results we've discussed are from lab studies, cells and dishes, computer models.
Not yet tested in people or even whole animal models extensively.
Correct. We don't know yet how these compounds would behave in a complex living system. Safety, effectiveness in a whole organism, those are still big unknowns.
So a lot more research is needed before this could potentially reach patients.
A huge amount. Extensive preclinical work, usually involving animal studies, is required first to look at safety and efficacy in a more realistic setting.
And if that goes well.
Then, and only then, would it move to clinical trials in humans. That whole process from lab bench to bedside is long, expensive, and unfortunately, many promising candidates don't make it all the way.
Years, typically.
Easily. And the researchers themselves point out there's more work to do on optimizing these molecules.
Like making them even better.
Yeah. Refining the chemical structure to potentially boost potency even further, maybe improve drug-like properties, and just generally understand the structure-activity relationship better. They even mentioned struggling to make one specific variation, compound 19F.
Ah, okay. So ongoing chemistry work is needed too.
Definitely. They're aware that these current leads, while exciting, likely aren't the final product.
It's good they're realistic about the next steps. The news article quoted Dr. Paul Foster, one of the researchers. What did he say?
He called it an exciting new development that could pave the way for treating the most common breast cancer when other therapies stop working.
So highlighting that potential for resistant cases.
Yes. He emphasized how these benzofuran molecules offer a different way to suppress estrogen and reduce tumor growth by hitting both enzymes.
And the article also mentioned future plans for him and his colleague, Dr. Claire Simons.
It did. They expressed hope that this work will lead to a new class of treatments and, importantly, said they are already working on developing even more potent dual inhibitors.
Actively pursuing improvements.
Exactly. Trying to build on these findings specifically to tackle that problem of treatment resistance.
Well, this has been a really fascinating look at some cutting-edge science. As we wrap up, what's a final thought for our listeners to take away from this?
Maybe just to reflect on the incredible complexity within our own bodies, the intricate dance of enzymes, and how understanding these pathways at a molecular level allows scientists to design these really clever strategies.
Like targeting multiple points at once.
Yeah. It shows the power of that deep biological understanding for finding new ways to tackle difficult diseases like cancer.
And if this has sparked your interest, listener, and you want to know more about hormone therapy resistance or drug discovery, there are great resources out there.
Reputable cancer research organizations have a wealth of information.
This has just been a glimpse into one specific piece of that ongoing effort. Thanks for joining us for this deep dive.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.